Well, aloha everyone, and welcome back to Human Nutrition here at Chaminade University. Today we'll be discussing carbohydrates, which will include sugars, starches, and fibers. First, we'll talk about the difference between whole versus refined grains, and then we'll discuss examples of monosaccharides and disaccharides. We'll discuss how monosaccharides are absorbed in the small intestines. We'll have to break them down from disaccharides to do that. And then we'll talk about the role of insulin and glucagon in the regulation of your blood sugar levels, which is very important. So, most modern diets rely heavily on carbohydrates. And if you find carbohydrates in whole foods, in foods that are unadulterated, you find them in their most natural state. And when we tend to have processed foods, that means that we've refined the carbohydrates. Typically, when we do this, we're going to separate them from their vitamins, their minerals, and their fibers. So while we will still have calories, we will have much less nutrient-dense calories. Now, you can find carbohydrates in almost every one of the food groups, with the exception being the oils and fats. So we can find it in the proteins and things like seeds and beans and peanuts. In the dairy, we can find it in whole milk. 1% milk, and in low-fat yogurt. We can also find it in fruits and in vegetables and, of course, in grains. Now, when we talk about whole versus refined, we mean, have we changed it from its native state, typically in order to preserve it, but also sometimes in order to more easily be able to digest it? This would be something that would be unrefined. So the example would be corn on the cob. Now, if we were to take that and turn it into corn flakes, we will have refined it, and we will have removed many parts of the corn to be able to do that. Oftentimes, we'll be removing some of the most nutritious parts. Another example here is unrefined sugar. Here's sugar cane, and this is what the refined table sugar, or sucrose, that we see when we sit down at a restaurant to put into our coffee, for example. If we're looking at a grain, a whole grain is going to include multiple parts. So the outermost layer is called the bran layer. There's a lot of fiber found in the bran. It's a very good source of vitamins and minerals. We also have the germ, which is the embryo area where sprouting is going to occur if this were to fall on the ground and turn into a plant. It's really high in oils and really high in vitamin E. And the endosperm is the largest part of the kernel internally here. It's made up mainly of starch, but it also is going to contain some protein and some vitamins and minerals. Now, when we refine those grains, we generally just use the endosperm, just this part. And that means that it's mainly going to be starch, and we've lost all of the nutrition in the bran and in the germ. Now, refined grains can often be enriched, so they'll take the vitamins away and then they'll add the vitamins back, if that makes sense. But they often will enrich them with things that we generally don't get enough of in our diet. That will include things like thiamine, riboflavin, niacin, and iron. And they also fortify them with other things like folates. Typically, however, they're still going to be missing some of the things that are found in whole grains. That's going to include things like magnesium, vitamin E, and multiple B vitamins. Now this here is just demonstrating, and you may have heard me say this in multiple little lectures before, as a side comment at least, but a lot of times when nutritionists start to evaluate someone's diet, they recognize that a lot of the sugars are hidden sugars that are found in their drinks. Now here's an example of eating some kiwi versus drinking a soda. Now while you get about the same amount of energy in terms of your calories, right? That means you're going to get approximately the same amount of carbohydrates or sugars. But nutritionally, the soda is very nutritionally poor, and the kiwi is much more nutritionally dense, right? The kiwi has a high vitamin C, folate, and calcium, and there's very little of that found in the sodas. This diagram depicts the process of photosynthesis. Now, photosynthesis is something that is unique to plants, and of course some algae. Basically what happens with plants is that they're going to take carbon dioxide and water, literally air and water, and they're going to convert the energy of the sun into all of the biomass of this plant, basically by converting it into glucose and then into starch. This is also going to off-gas oxygen. So they take in carbon dioxide and water, and they off-gas oxygen. As a side note, this is why they are very important in terms of greenhouse gas emissions, because they're going to help offset those greenhouse gases by taking that carbon dioxide, making themselves a carbon sink, and releasing the oxygen that we as humans are going to need to respire. Now, plants are going to take that glucose and convert it into a starch. And when we eat the plants, or if we eat an animal that has eaten the plant, etc., the digestion is going to take that starch that they've converted from glucose and going to kind of break it down and convert it back into glucose from the starch. Now here's an overview of what I just said about photosynthesis, right? So we bring in carbon dioxide and we bring in water, and we create sugar, this is glucose, and we off-gas oxygen. We also are going to off-gas water, but because we put more water in than water out, we can kind of eliminate that. So oftentimes you don't see this water depicted. You just see that we are going to take up 6 water molecules. But my point is that every time that we take that carbon dioxide out of the air, we turn it into the biomass of the plant. So in this way, the plant becomes a carbon sink and takes the carbon out of our atmosphere, right, which is good for carbon emissions, etc. And also, it's going to off-gas oxygen, which is going to help recycle and help clean our air so that we as human beings are able to respire it. Now let's talk about monosaccharides versus disaccharides. As you may remember, anytime that we have a biomolecule, it's generally going to be comprised of, there's a monomeric form and a dimeric form, and the dimeric form is comprised of 2 monomers, right? And you could also have, say, a trimeric form, or above that, just a polymeric form, right? Called like an oligosaccharide, for example. So anyway, monosaccharides, when we have 1 monomer of a sugar, so a monomeric sugar molecule. And glucose is an example of monosaccharides. Now glucose here, it's a 6-carbon chain, turns into a ring. Fructose is another example of a monosaccharide. This is often found in fruits, vegetables. You have heard of it of high fructose corn syrup. It's often added to things as a sweetener. And again, it's going to be a 6-carbon molecule, 2, 3, 4, 5, 6, in a ring, but it's going to be arranged in a different conformation. Galactose is another example of a monosaccharide. It's also going to be found in milk. So this is something that you will see again when we talk about lactose tolerance. Galactose is the monosaccharide that is part of the milk sugar, lactose. So for example, we were just talking about that. Here's lactose itself. It's made of galactose and glucose. Now this would be an example of a disaccharide. A disaccharide is when we have a dimer of sugar, right? A dimeric sugar molecule. And that can be comprised of many combinations of the different monomers. In this case, maltose would be glucose and glucose. We also can have glucose and fructose come together to make sucrose, and sucrose is what we know as table sugar, so this is what you're often going to recognize when you think of sugar, but it's actually a dimer or a disaccharide. And again, lactose is going to be milk sugar found in milk, ice cream, and other dairy products. Incidentally, if you have an issue with lactose digestion, you can take something called Lactaid. Oftentimes, the reason that you're having issues with digestion of lactose is low levels or no levels of the enzyme called lactase. Remember, anytime you see ACE, that indicates an enzyme, so lactase is going to break down lactose. So if you don't have enough lactase in your system and you have an intolerance to lactose, you can always take Lactaid, which would be an exogenous form of the enzyme that you don't actually produce, and then you can go ahead and enjoy that ice cream. This slide depicts chemical reactions that are going to happen when we are taking either a dimer and breaking it into 2 monomers, so dimer being 2, breaking down into 2 individual, right? So this is the di and then monos. On this side, we would have a condensation reaction. You may have heard of this as a dehydration synthesis reaction. This is when we have 2 monomers that are going to come together and form a dimer. And this is how all monomeric and dimeric synthesis happens. Basically what happens is that we have a water molecule that's going to be split in half. That's why it's called hydrolysis, And again, this is in the case of taking a dimer, which is already a package of 2, and breaking it into 2 packages of 1. We split that water molecule in half, and we use that to cap the ends that occur when we slice through here. So we have the OH that's going to cap off and make that carbon happy. This H is going to make that oxygen happy. And that means that both of these monomers are going to become less reactive, right? When these monomers are going to come together, down here in this case, then we're going to have to chop that OH group and H group off. And then they're going to form water. That's called a condensation reaction. You may also have heard of it as a dehydration synthesis reaction. Again, that's when we're taking 2 monomers, putting them together into 1 dimer, and to do that we're cleaving off the equivalent of 1 water molecule. Now when we talk about a complex carbohydrate, it's often way longer than a disaccharide. So we just talked about a disaccharide, which would be just 2 of these monomeric forms of sugars. But oligosaccharides are going to be a shorter chain, but longer than 2, so up to 10. Once we get to 10 and more, that's called a polysaccharide. These are very long chains of monosaccharides. And typically this is going to include the starch and fiber that are found in plants. Also, it's going to include the glycogen that's found in animals. It's found like in the animal liver and in the muscle, for example. So these are examples of polysaccharides. And as you can see, although they are comprised of different monomeric subunits, the overall shape is quite different. So you can basically take these little Lego pieces and make very complex shapes out of them. And the difference in the shape is going to be a major difference between, say, cellulose, starch, and glycogen. Now, cellulose is something that is indigestible to humans. Incidentally, it is digestible to termites. That's why they're able to eat trees. But we are able to digest starch that we get from plants and glycogen that we can get from animals. And all of those are going to give us smaller subunits of glucose once we break them off, right? And then we can use that for the processes of metabolism. And we'll talk about that in just a moment. But let's talk about another form of these long polysaccharides, including fiber. So soluble fiber can be found in multiple different foods. And soluble fiber is different from insoluble fiber in that soluble fiber is going to be what we call solubilized or go into solution fairly well. Insoluble fiber is not going to go into solution very well. Soluble fiber can be found in things like oat bran, barley, multiple different nuts, fruits, vegetables, etc. Insoluble fiber is found in whole wheat, whole grain products, vegetables, and wheat bran. And these are going to play different roles in your body in terms of whether or not they have nutritional content. Now, while insoluble fiber has no nutritional content, it's definitely going to have content or assist in your digestive system, and we'll talk about how that works in a little bit. Now, fiber is going to include complex carbohydrates that we're not able to digest, and that means that we are not able to digest it or absorb it. And again, the difference between soluble and insoluble fiber is whether or not it's going to be able to go into solution. Does it dissolve in water or not? And oftentimes they'll use fibers to add to processed foods in order to thicken foods and reduce fat or calories in the foods. An example of fiber here would be pectin. And obviously I would never quiz you on what these actually look like on the exam. I just wanted to throw that up for you. Now, when we talk about the digestion of each of these biomolecules, we'll talk about where they start and the different versions of the biomolecule that are found at different stages of digestion. So let's start in the mouth. Now, salivary amylase is one of the first enzymes that's going to be secreted. Again, it's secreted in the mouth, and it's going to start breaking down those carbohydrates, hopefully into polysaccharides. Now, that they're not going to have much digestion in the stomach. Stomach's going to be digesting mainly proteins. But in the small intestines, we have another amylase that's going to be secreted by the pancreas. Remember, the pancreas is going to be secreting digestive enzymes into the small intestines to help bring that pH level back to normal from the stomach, which has an extremely low pH relative to the small intestines. And amylase, pancreatic amylase, is going to work at that more neutral pH. So it's going to wait until we have got that chime back to a pH of about 7, and then it'll help digest those polysaccharides into monosaccharides like glucose, galactose, and fructose. Now, these are going to get picked up by a process of active transport and brought into the bloodstream in the form of monosaccharides through the intestinal lining. So you can see we've taken long chain sugars, broken them down into shorter chain sugars, even shorter chain sugars, and eventually down into monosaccharides, which are not a chain at all, right? Individual Lego pieces. And again, those are what are going to get picked up and carried about through the body by the bloodstream. So let's talk about how they're absorbed across the intestinal lining. So this would be the lumen of the small intestines. These would be the epithelial cells of the small intestines, and then this would be the villi core. Now, as you may remember, we have villi at the tip of these. We have microvilli with little brush border enzymes on them, etc. And this is where we're going to be able to pick up the carbohydrates. So that happens by way of a sodium glucose transport proteins. It's these guys here. And basically what happens is we have glucose and fructose and galactose coming in, and they're going to get transported in along with sodium. So sodium's also going to get pulled in. And then on the other side, I'll just follow the sodium pathway for a second. So sodium gets pulled in here, and then sodium is going to get pumped out into the core of the villus, and potassium is going to get pumped in. In this way, we're going to maintain the correct pH, etc. So we're not going to have any sort of odd change in charge from one side to the other, but what we're going to end up with is a lack of sodium, which is going to pull in more sodium, which is then going to pull in more sugars, right? So as those sugars get pulled in, then they're going to get sent across this epithelial cell lining into the core of the villus by the process of facilitated diffusion. Whereas this one over here, the sodium-potassium, is going to be via active transport. And that's how we're going to be absorbing our monosaccharides across the intestinal lining to be able to be sent up for processing through the bloodstream. They're going to head over to the liver, and then they're going to end up being pumped back into the general blood population. Again, as I mentioned, lactose is a milk sugar, and you need a specific enzyme to digest that. You'll need the enzyme lactase. And if we're unable to digest lactose, and lactose ends up in the digestive tract, it's going to end up passing through the small intestines undigested and into the large intestines. And this can cause serious problems because in the large intestines, it's really only supposed to be picking up water and other minerals, etc. So we're not going to be absorbing anything in the large intestines, which means that the bacteria in the large intestines are going to have extra food to feast on. And that can cause all sorts of digestive issues. And if the undigested lactose is in the small intestines, it can cause abdominal cramping and distension and ultimately end in diarrhea. So here's what normal lactose digestion looks like. So lactose is meant to be broken down in the small intestines by an enzyme named lactase. Now remember, lactose is comprised of glucose and galactose, so once it's broken down by lactase, both glucose and galactose can jump ship and end up into the capillaries on the other side, and here in the intestinal lumen. Now, that results in normal passage of your stools. Now, if we have an issue where we have no lactase, right? So we're not going to break down glucose and galactose. That means the bacteria are going to be able to feast on that lactose, which can cause gases and other osmotically active molecules to cause all sorts of issues, including increased motility, like movements, and also irritation. And obviously, this can cause irritability in the individual. What's interesting about lactose intolerance is that it's one of those things that's highly lauded as being a positive mutation. Now, if you were to picture that some individuals would die during childbirth, and then there would be a small population of infants that did not have access to mother's breast milk. If those infants had access to cow milk and were able to digest the cow milk, they had a much greater chance of surviving than the individuals who had access to cow milk but were unable to digest it. So once a mutation arose in a population, it spread very quickly and became pervasive. And so it's interesting to actually see how lactose intolerance, it actually lines up with borders of countries and etc. And you can see areas, this is where we have the highest lactose intolerance, and then the least amount of lactose intolerance means these people have high amounts of lactase. And again, when you see we have exceptions in certain populations, you can see they're of different descent, like the American Indians and the African Americans, which would come from these lineages, and the Australian Aborigines. Again, they would have been native to Australia, but they wouldn't have had necessarily the same opportunity to have that mutation. Anyway, that's a map of worldwide lactose intolerance. Now, we have indigestible carbohydrates, such as starches that are resistant to digestion, fiber, as I mentioned previously, and oligosaccharides that are unable to be digested by the small intestines. And these are going to affect the amount of time that stool takes to transit, the type of intestinal microflora that is found in the different regions of the intestines, and that in turn can help the amount of, that in turn can be correlated with the amount of intestinal gas that's created. All of this can also affect nutrient absorption rates. So depending on what you have in there to absorb and what type of microflora are available to be able to absorb it or to be able to break it down so you're able to absorb it, this can obviously affect the amount of nutrients that are absorbed. Now, this is an example of how indigestible carbohydrates, such as fibers, are able to slow down nutrient absorption. Now here, if we're in a fiber-rich meal versus a low-fiber meal, right, the fiber-rich meal is going to pull in more water, which is going to make this slow down a little bit. Remember, just a little bit is going to be released from the stomach at a time. And that means that it's going to have more of a transit time. A longer transit time means that we're able to allow for absorption throughout the entire range of the small intestines, as opposed to in just a small region, right? In a low fiber meal, everything's going to be smaller amounts of volume because it's not going to pull in that amount of water. And then they're all going to be trying to be absorbed in this upper region of the small intestines instead of throughout the entire tract. So indigestible carbohydrates are very helpful because they're going to allow you to utilize the entire small intestines to be able to have nutrient absorption occur. All right, so let's talk about blood glucose levels. So we can get and store glucose in the form of glycogen in the muscle, in the form of glycogen in the liver, and also in the form of glucose just as glucose in fat-storing cells. So these are all places where we have reservoirs of glucose in the body. Now, the pancreas secretes insulin, and insulin affects all of these pathways. So if we have an increase in blood glucose, that leads to an increase in the insulin secreted by the pancreas. And that means that we're able to take that glucose. Insulin's going to affect the rate at which glucose is stored as glycogen in the muscle and in the liver, and also the rate at which glucose is stored in the fat. When it does that, it also is going to mean that we're going to be reducing the amount of blood glucose, right? So that's going to return us back to the normal range of blood glucose. Now, if we have decreased blood glucose, we're going to start at the bottom here. That means that the pancreas is going to secrete glucagon. Glucagon is going to have opposite effects here in the liver. It's going to produce glucose both from the breakdown of glycogen, or glycogenolysis, and the creation of new glucose, or gluconeogenesis, from glucose precursor molecules. Both of these are going to result in the increased amount of glucose released into the blood, restoring the range of blood glucose back to normal. Right, and so this graph here shows blood glucose levels on the left and time after eating, on the x-axis here, so y and x-axis. And as you can see, immediately after eating, blood glucose is going to, I don't want to say skyrocket, but it's going to increase. That's going to cause insulin to be released, which is going to stimulate both the uptake and the storage of glucose, which means that we're going to fall. Right? So several hours after a meal, the blood glucose level is going to drop. And if it drops too low, right, sometimes it falls underneath the fasting level of blood glucose, which would be where you would expect your blood glucose to be after restoring it to normal. And that means that we're going to activate this pathway where pancreas is going to secrete glucagon. Again, that's going to release glucose in the form of breaking down glycogen and creating new glucose. All of that's going to restore a normal range of blood glucose, which you can see there. All right, so in order to provide glucose in a steady supply to the cell, we have to regulate the concentration of glucose by the enzymes that are secreted by the pancreas. Okay, so we have what's called, and the liver is involved in this as well. We have what's called a glycemic response, which is basically how quickly your blood glucose is going to rise and how high your blood glucose is going to rise after carbohydrates are consumed. Glycemic response. How fast and how sharp is this curve? Does it stop around 125, or does it spike really high, or does it spike even higher, right? Pre-diabetic or diabetic conditions. Now, the glycemic index is basically how different foods are going to affect your glycemic response. And if you wanted to calculate out your glycemic load, you would calculate the glycemic index by the amount of carbohydrates that are found in one serving of that food. Now, this image shows foods that have a low glycemic index, a medium glycemic index, and a high glycemic index, as well as, low glycemic index, medium glycemic index, high glycemic index, on the top, as well as the low glycemic load, medium glycemic load, and high glycemic load. And if you want something that was low in both the glycemic index and the glycemic load, you would choose something like apples, carrots, etc. Or high glycemic load would be something, and a high glycemic index is something like baked russet potatoes or cornflakes. And you can have kind of multiple different combinations in between as well. So depending on how you respond to sugar, you can determine what you want to eat in terms of the glycemic load that you're putting into the body. Okay, so previously I talked about the fact that sugar was used as energy. So in order to create energy, glucose is going to be metabolized or broken down through the process of cellular respiration. That's also called aerobic respiration or aerobic metabolism because we do use oxygen. There's another form called anaerobic respiration, and that results in far less bang for your buck. You get way less ATP out of anaerobic respiration. So this cellular respiration, again, it's aerobic, produces ATP. And basically what happens is we're going to take one molecule of glucose, I apologize, and add 6 molecules of oxygen, and we're going to break that molecule of glucose into 6 carbon dioxide molecules. So we're basically taking one 6-carbon molecule and turning it into 6 1-carbon molecules, also creating molecules of water, and at the end of this pathway creating 38 molecules of ATP. So one molecule of glucose is going to give 38 molecules of ATP. Now remember, ATP is used as energy currency throughout the cell. So how does it do that? All right, so the very first step is that we take that 6-carbon sugar, glucose, and we break it down into pyruvate. So we take one 6-carbon biomolecule and we make 2 3-carbon molecules out of it. And that's going to release a little bit of ATP. It's also going to release here an electron, and we're not going to get extensively into the electron transport chain. We'll just basically say when we get to the electron transport chain, that makes more ATP. All right, so we take those pyruvate molecules. Each of them are going to enter into the mitochondria. So just to be clear, this anaerobic metabolism up here, this would be what we would get without having oxygen in our pathway. So we would be able to break down glucose into pyruvate, and that would be it. We would release this ATP, and boom, that would be all we had. Instead, we're able to take that pyruvate molecule and pop it into a nice little organelle called the mitochondria, and the mitochondrion, singular, mitochondria plural, the mitochondrion is going to enter into the acetyl-CoA formation from pyruvate. That's going to result in the release of carbon dioxide and more electrons, but all of those are going to go into the electron transport chain. Eventually those are like, picture those like, instead of, if we're collecting dollars here, in terms of ATP, these electrons are coins, but eventually we'll add up, so we'll eventually cash those in later. Anyway, here we have 2 acetyl-CoA, so we have now gotten down to the point, because we've released those carbon molecules, carbon dioxide molecules, we've basically gotten that from here. So we've taken 2 3-carbon molecules, and now we've broken them down into 2 2-carbon molecules and released 2 carbon dioxide. These 2 carbon molecules are going to go into the citric acid cycle, where they're going to result in the release of 4 carbon dioxide. Again, as you can see, we have 2 2-carbons, now we have 4 1-carbon molecules. That's going to result in the release of ATP, and again, more electrons. Later on, we're going to cash those electrons in the form of ATP. And that is how we get energy out of a molecule of glucose. All right, so now we're going to talk about diabetes. And diabetes is basically what happens when your blood glucose levels are not responsive in normal ways. And there's multiple different ways that can happen. We'll talk about the 2 types of diabetes, and then we'll talk about how individuals can use a low carbohydrate diet to help manage their weight, and talk about how fiber is related to heart health and bowel disorders. And lastly, we'll talk about non-nutritive sweeteners. All right, so facilitated diffusion is different from active transport in that facilitated diffusion is still going to be going with the concentration gradient. So here we have a high concentration and we're going towards the low concentration. This is similar to regular diffusion, except that with regular diffusion you'd be able to go straight through the membrane. But with with facilitated diffusion, I apologize, you need a carrier molecule to assist with that. And here's active transport. So active transport is going to utilize molecules that are going to be going already with the concentration gradient, and then have a symporter, which means that it's going to take both molecules. So here it's taking sodium and glucose. It's called a symport transporter, meaning that they're both going in the same direction. It's going to bring glucose into the cytoplasm. So it's more complicated because we have an ion gradient. That ion gradient is created here at step 1, where sodium is going to be pumped out and potassium is pumped in. And in that way, we have an ion gradient where we don't necessarily have a charge gradient because they're both positively charged molecules, so we're swapping one positive for one positive, but that means that we have a gradient of sodium that's higher on the outside, and we're going to piggyback on this gradient of sodium, which needs to come back in, And in that way, we're going to pull glucose in as well via the symport molecules. So sodium goes out alone in exchange for potassium sneaking in, and then sodium comes back in with its new friend glucose to bring glucose into the cytoplasm. Again, that's called symport mechanisms. All right, so we talked about how sugars are tightly linked to your health. We're going to talk about diabetes and hypoglycemia, and then also dental caries or dental disease. We'll talk about how that also has an effect on weight management, heart disease, and bowel health. Now, as we saw earlier, this is our normal response in terms of our glycemic response to sugar. So here we are having eaten nothing. Now we have an introduction of sugar. Over the next hour, hour and a half, we're going to have a spike to that sugar. That's going to kick insulin on. When insulin kicks on, we're going to have a drop in that sugar. It might drop too far, and if it does, we'll have to restore it back up. And that's normal conditions. Now, pre-diabetic would mean that we have a heck of a spike when we introduce sugar to our system. But then insulin does still kind of get it back under control. It takes a little bit longer and it bounces a little bit too far. So this is a pathway that is failing. It has not failed yet. It still gets us back to its final result, but it takes longer because it peaks higher and then it oscillates further down before it gets it back under control. So pre-diabetic conditions means you might still want to be monitoring your blood sugar because both of these conditions can make you feel kind of sick. This is a diabetic condition. Now, I want to point off that, first of all, they start off at a higher baseline by far, at a baseline that's higher than a spike in a normal individual, and is almost as high as the spike in the pre-diabetic individual. And when they have sugar introduced to their body, they spike way high. And either they have a no response or an abnormal response to insulin, so they take a very long time to restore back down. And even then, they don't even restore back down to their own level in the 6-hour time frame, whereas the normal individual has been restored back to their normal glucose levels around 3 hours. Okay, so we have two different types of diabetes. Basically, diabetes is when we either have a lack of insulin or we have a lack of insulin response. So type 1 diabetes means insulin is no longer made in the body. And while this means that this individual always has to check their blood sugar and always have to make sure they have insulin on them, which can be extraordinarily expensive because insulin is a very expensive medicine. But on top of that, well, I guess my point is that they are going to be able to have a response to insulin. So even though it seems awful because you always have to carry it with you, you're still able to have a response to insulin. Now, type 2 diabetes is when insulin is present, but the cells don't respond to that insulin. And this is the type of diabetes that can have pre-diabetic conditions as well, whereby they do respond, but they respond more slowly, and then they finally do respond, and then they over-respond. So we can have an intermediate step between non-diabetic and diabetic conditions that are pre-diabetic conditions, whereby we are still getting an insulin response, but it's not adequate. Now, immediate symptoms of diabetes include excessive thirst, which results in frequent urination, right? We're going to have issues with salt balance as well. We're also going to have issues with blurred vision and weight loss. Long-term complications can include vascular damage, heart damage, damage to your kidneys. Again, that blurred vision can turn into long-term vision damage, and your nervous system. Additionally, these individuals are less able to fight off infections. They have more of a lowered immune response, and that means that they can oftentimes have sores that take a very long time to heal, never heal at all, or result in amputations. Oftentimes, once they have that first amputation, they'll take off a couple toes. Very soon after, within the next couple of years at best, they'll be taking off a little bit further, perhaps up to the foot and then to the knee, etc. So once you make that first amputation, you're pretty much headed down a path where, although we have staved off the long-term removal of that leg or that limb, it is still going to be occurring in the near future. Okay, so this is going to include some complications, and complications are going to include damage to blood vessels, which can lead to multiple things, including risk of stroke, high blood pressure, risk of heart attack, or the arteries can be blocked in the legs, which can then, of course, lead to a risk of stroke and heart attack. Can cause reduced blood flow to the feet, and because This is going to cause reduced blood flow. That means we can have sores, and the sores aren't going to be getting the proper healing compounds from the immune system because the blood flow isn't delivering them very quickly. And because they already have high glucose levels, that means that microbes really like their blood, which can lead to lesions. Additionally, because we can have nerve damage in these individuals, that means that we can also have increased risk of accidental injury, which can open sores that are going to take a while to heal. On top of that, because we have nerve damage, these individuals may not be able to feel the sores, which means they may neglect treatment or even exacerbate the condition by scratching or whatnot because they can't actually feel the pain. Um, some other damage can include damage to the kidneys, which can cause protein excretion in the urine and eventually cause renal failure. Additionally, that can cause the small blood vessel bleeding, can cause bleeding in the retina, which can lead to blindness. So obviously we want to try to manage this condition and try to keep it as much under control as possible. And the goal of diabetes treatment is threefold. We're basically going to be using diet, medication, and exercise to try to maintain normal blood glucose levels in these individuals. So we can use diet that needs to be very carefully planned so that the blood glucose is going to have sustained conditions. That means we can never give too many carbohydrates, but again, we also have to have carbohydrates on hand at any point if the glucose levels fall too low. Exercise can help control blood glucose in some ways, and that can help offset the amount of medication required or the having to stick to a very strict diet. But most of these individuals are still going to be reliant on medication. Typically, I mean, everyone that has type 1 diabetes is reliant on insulin. And in type 2 diabetes, glucose levels are able to be managed by different oral medications as well as insulin. Now remember, in pre-diabetic conditions, we do still have some insulin response, so we could just have to give these individuals more insulin. But individuals that don't have a response to insulin might respond to different oral medications. Okay, so what is hypoglycemia? Now, hypoglycemia can be described as two conditions: one, fasting, and two, reactive. Now, reactive hypoglycemia is when we have too much insulin secreted when carbohydrates are consumed. And in order to help offset this, individuals typically have to eat a ton of protein during every meal and have many smaller meals so that they're never given too much sugar at once, so they never have a super overreaction in this insulin response. Now, fasting hypoglycemia, and again, hypoglycemia is low blood sugar, can occur if we have abnormal insulin secretion that's not related to food intake. And hypoglycemia can result in sweating, blurry vision, dizziness, anxiety, hunger, weakness, fatigue. Typically, this is one of the very first things that they'll feel. They'll feel very fatigued, and it'll end up with shakiness, fast heartbeat, headache, and obviously irritability, all these symptoms. And that's why these individuals always have to make sure that they have carbohydrate sources on hand, right? Just a glass of fruit juice or something like that to help bring their sugar levels back up immediately if they end up falling too low, because they might have an overreaction to the insulin response. So dental caries are also something that people who overeat carbohydrates and then have bad dental health can face. So if we have sugar, which is found, sucrose is basically going to be the result of breaking down carbohydrates. So if you eat a piece of toast and then you leave it in your mouth and little pieces of toast end up in between your teeth, and then it gets broken down by salivary amylase into sucrose, and then sucrose is going to interact with that bacteria. That bacteria can lead to the formation of plaques and the production of acid, and that can cause the tooth enamel to have decay. And once we have tooth enamel decay, then we end up with a little pocket. So here's our enamel. If we end up with a pocket in that enamel, a little small hole, the acid can continue to dissolve that enamel, and typically the individual doesn't even know it in this case. So if you catch it at this case and you can seal that up at your dentist, you're typically pretty good. But if dental decay reaches inside the dentine layer, now this becomes very painful, and it's going to travel very quickly because the enamel is quite tough to get through. But this dentine layer is a little bit softer, and eventually it's going to reach the pulp layer. And when it reaches the pulp layer and into the root, this can be very painful. It can cause abscess, etc., and this is a tooth that's going to need extraction. Okay, so oftentimes individuals who want to manage their weight go through what's called the keto diets, right? Low carbohydrate diets. Here's an example of the keto food pyramid: 65% fat, 30% proteins, some green leafy vegetables to get your fiber in, and also some of your nutrients, and almost no sugars at all if possible. Now, the idea with this is that if we have carbohydrates in our system, that stimulates the release of insulin, which is true. And insulin promotes energy storage. That means that it's going to be taking energy and storing it in fat. And so the more insulin that you release, the more fat that you store, which means that a low carbohydrate diet can cause you to have less insulin released and therefore less fat stored. Now, if we enter into a low carbohydrate diet, we're going to create ketones, and ketones are known to help suppress the appetite. So once you get to the point that you have gotten through the first day or so, and you're starting to produce ketones, then you're going to have appetite suppression, and that at that point the diet becomes a little bit easier to maintain. So let's talk about carbohydrates and heart disease. So here we have soluble fiber shown here and cholesterol shown there. Now if we get soluble fiber, and again we can get that from oats, etc., then that's going to help take the cholesterol out of our system, right? It's going to form a gel with the cholesterol, and that cholesterol is also going to be coming from the liver as well. And it's going to trap it and take it out of the body, which helps prevent it from being reabsorbed back into the blood. So it's going to help remove the cholesterol from your body. Now, water-soluble fiber, again, it's going to bind dietary cholesterol, and that's going to help reduce the absorption of cholesterol back into the system. Fiber is also related to bowel disorders. Diets high in fiber and indigestible carbohydrates are able to help offset or perhaps prevent bowel disorders such as hemorrhoids, diverticulitis, diverticulosis, and constipation. These here are little diverticula, which are little pockets in the intestines where food or fecal matter is just going to get trapped and not actually move through. And if you can picture that the fiber acts kind of like brooms, it's going to help sweep through all these pockets and pull out matter that might not have moved through previously. We've also been able to relate fiber intake to colon cancer, because if we have high diets in fiber, the populations that have high diets that are high in fiber, have a lower incidence of colon cancer. However, intervention does not support that study, so once it's too late, it's too late. Now here's an example of how colon cancer is going to be created and then metastasize. So here's normal colon epithelium. Here's one mutated cell which starts to grow and divide. It becomes a tumor. It enters into additional mutations and becomes a large tumor, which eventually ends up malignant. Now, if that large tumor you can see actually has two populations of cells, this population has undergone its second mutation and has become malignant, and it's going to break out from that normal colon epithelium and extravate into the blood vessels, where they're going to be able to what's called metastasize, or move to and implant in other regions of the body. Eventually bone, probably, and brain. So how do we assess our carbohydrate intake? And we talked about this previously when we talked about the FDA food label. Basically, in order to determine the percentage of energy that comes from your carbohydrates, you need to determine two things first: the total amount of energy in kcals for the day, and the amount of grams of carbohydrates in the day's diet. Once you know the amount of grams of carbohydrates, you're going to multiply that by 4. Remember, every gram of carbohydrates gives you 4 kcal. If it were fat, we would be using 9, right? So each one of these different biomolecules has a different kcal per gram. Anyway, for carbohydrates, it's 4, so you're going to multiply it by 4 to get the amount of kcals those carbohydrates provided. And then you're going to be able to figure out the amount of energy that day that came from those carbohydrates by taking the grams of carbohydrates times 4, whatever that number was, and dividing it by the amount of grams of carbohydrates that were your entire for the day, right? So if we take the amount of energy from the carbohydrates and we divide it by the total amount of energy, and you multiply it by 100 to make it a percentage, that's going to give you the total amount of energy from the carbohydrates for your daily diet. Now here's an example. If a diet has 2,500 kcals and 350 grams of carbohydrates, you take that 350 and multiply it by 4 to get the total kcals from carbohydrates for the day. That's 1,400. You take that 1,400. Well, you take 2,500 and divide it by that 1,400. And that's going to be 56% in this case of kcals came from carbohydrates. So how do you choose healthy carbohydrates, right? If you're going to have 50% of your diet or so coming from carbohydrates, you want to make sure that you choose your carbohydrates wisely. You want to make sure that half or more of your grains come from whole grains. Whole wheat, oat bran, rye, pumpernickel bread. Use whole wheat pasta. Use brown rice. Um, Use plain oatmeal and use raisins or other types of fruit for sweetness. Make sure that you check all your ingredient lists to see the word whole or whole grain before whatever the grain ingredient's name is. And always try to increase your fruits and vegetables. Make sure you add in your beans, such as kidney beans, chickpeas, black beans, etc. They have tons more fiber and resistant starch than other vegetables, and that fiber is going to be really helpful, again, as a broom to help clean out some of those pockets in the intestines and help avoid intestinal polyps. We want to add things like berries and bananas and dried fruit to cereals and desserts. So if you need a sweetener, try to choose fruit sugars if possible, and natural fruit sugars, not high fructose corn syrup. Make sure that you eat a ton of veggies, lettuce, tomato, etc. on your sandwich. And have a vegetable at dinner as well, maybe two, and try a new fruit. Every time that you introduce new sources of carbohydrates like fruits, you're going to also be introducing a variety of nutrients that might not have been provided from your other sources. Limit the amount of sugars that come from added sugars. And if that means that you're going to have to drink less soda, right, 12 instead of 20 ounce, or you can switch to water or low-fat milk instead. If you're baking yourself, try to use less sugar in your recipes and use fruit instead of candy or other types of sweets. And again, always check the ingredients list on everything to try to make sure the ones are the healthiest for you, in this case, the ones that have the least amount of added sugar. We also have several examples of non-nutritive sweeteners. That's going to include things like saccharin, aspartame, and sulfamine K. So if you see these in your ingredients list, you know that you're dealing with non-nutritive sweeteners. What do I mean by that? I mean it's something that causes your tongue to taste sweet, so you get that sweet response in your gustatory cells, but you're not going to have nutrition from it. So it's not going to break down into any calories. And when they say 300 times sweeter, they really mean that you need 1/300th of the dose to be able to induce the response in the gustatory cells. Aspartame is another example. It's approximately 200 times quote-unquote sweeter, and sulfamine K is 200 times sweeter as well. Again, I'll never ever give you chemical images on the exam for you to have to know. Just recognize that these are things that trick the body into thinking that it's sweet without having any nutrition from it, because it doesn't break down and turn into any sort of energy for the body. Another example is sucralose. We also have ribena. Sucralose is a version of sucrose that is modified such that we cannot digest it. Right, so here's sucralose and here's sucrose. So you can see we have these modifications, these little chlorides. So instead of the hydroxyl groups, or here instead of the hydrogen. So anyway, my point is it's changed a little bit, so it tastes sweet, but we can't break it down, which means we can't enter it into the cellular respiration, which means we can't use it for energy. We also have ribena, which is going to come from the stevia plant, and this is going to be approximately 300 times sweeter than sucrose. Okay, everyone, thank you so very much for listening to my lecture today. I appreciate it. Well, aloha and happy studying. Have a great day.